Welcome to the Everything Epigenetics podcast, where we discuss DNA regulation and the insights it can tell you about your health. I'm Hannah Went, and I'm the founder of Everything Epigenetics. Today, my guest is Dr. Toinét Cronjé. We are going to be discussing epigenetics and epidemiology. I'm really excited about this one. We're going to dive into what can epigenetics actually do for epidemiology, and why should epidemiologists start to take this data and put it into practical use? A quick introduction to my guest. Toinét Cronjé is an epidemiologist and postdoctoral researcher at the University of Copenhagen's Department of Public Health. She completed her doctoral training at the Center of Excellence for Nutrition at the Northwest University in South Africa, where she investigated the associations of individual and composite DNA methylation markers with cardiometabolic disease risk factors in an African population. She actually specializes in studying underserved populations, which adds a really unique aspect of research into this epigenetic field. Her current focus is a little different. She is looking and working with biomarker discovery in Alzheimer's disease and related dementias using multi-omic data sets. This work serves her overarching career goal of identifying biomarkers of complex disease risk and progression that are feasible to implement on a global scale. We'll talk more about her future work at the end of this episode too. And now for my guest, Dr. Toinét Cronjé. Welcome to the Everything Epigenetics podcast, Dr. Cronjé. I'm excited to have you here today. Thanks for being here.
Thank you so much. It's my pleasure to be here, and it's really nice to meet you.
Yeah, and you have a really unique background I absolutely want to hear more about. And I think our listeners would be excited to hear more about your journey and where you got your start and how you ended up where you are now. So would you mind giving us a little bit of insight into that?
Yeah, sure. It's a story of a lot of good mentors and luck. I was born in South Africa. I grew up there, did all my studies there. And what started out as a focus on psychology and nutrition moved into postgraduate work focusing on nutrition and cardiovascular disease. I got some interest in genetics during that time, and that evolved into epigenetics in my PhD thesis. And interestingly, I was visiting Copenhagen right at the end of my PhD thesis, and COVID lockdown happened, and I got stuck here, and they changed the visa guidelines. So I was allowed to apply for a postdoctoral position here, and I got one at the University of Copenhagen. And so now I'm moving a bit more into the proteomics research area. So overall, the golden thread is epidemiology. I'm an epidemiologist. I'm looking at noncommunicable diseases. And that is such a wide field. And we will probably talk about that a bit later. But I think the exciting thing about being a researcher right now, especially someone newly entering the world of science and research, is just how incredibly fast-paced it is, and how you can really jump from one thing to the next and explore everything. You can connect with people all over the world, even from a small town in South Africa. You can collaborate, you can do exciting research, and yeah, that is how I got here.
Yeah, that's great. I always talk about how amazing it is to have those really great mentors or those connections, especially in this research space, because it's not possible for an individual to keep up on all of the research that's being released, right? I think that's one of my goals of this podcast, Everything Epigenetics, is to try and bring people together so we can learn a little bit more about what's out there. And maybe that leads to collaboration on very unique data sets or looking at, particular diseases as well. So I definitely hear you on that. And I know you mentioned on your LinkedIn that you're an omic epidemiology enthusiast. I love that. But I want to dig into that. What are you studying now? If you could just explain a little bit of your postdoctoral work. I know you mentioned at the University of Copenhagen and what you're currently researching.
So epidemiology is just the term that encompasses investigation of humans, of populations. So in contrast to a lot of the people I talk to around here every day, I'm not a microbiologist or a drug developer, but I look at humans, at individuals in their fullness, and that encompasses everything from the molecular parts and the epigenome to the exposures around them and how that influences their epigenome, for example, or the other omics, and then how that relates to risk during their life course. And one of the very interesting things about Denmark, and why it is potentially a very interesting place to do research, is the availability of archived samples. It is all tissues that's been collected for the past 3-4 decades that they keep stored. There's a single linkage system for all the health records. And of course, with the appropriate data access and the ethics that goes into it, the idea is that you can now have these samples. You can go back to the biobank. I can find someone's liver sample from that time they had the biopsy before they started the treatment 20 years ago. And I know what has happened to them since because I have their electronic data. So the group that I'm in focuses on pathological biomarkers using biobank samples and different omics and imaging technology. And the idea is really to start comparing some of these methods. What does methylation tell us versus proteomics versus image-based AI, and what can be incorporated into clinical practice, and what is redundant, and what performs worse than what we currently have. So that's
That's great. I think we're going to see this field move in a multi-omic direction. Of course, I'm biased. I love epigenetics. But I will be the first person to admit they can't do everything, right? We have to look at every, angle, every omic, this idea of the multi-omic realm. And it's great that you all have so many access to those biobanks. I'm not an expert in biobanks whatsoever, but I feel like every time I talk to people who are in the Europe area somewhere, you all have so much access to them. I think that's something the US is lacking in, is kind of those biobank samples and that accessibility, which is crucial for doing the research that you're doing.
exactly, and especially when you're looking at a healthcare system that is so communal, where we're all going to the same place. We all have the same system. There's no private and non-private, and it's just, and it can all be linked. And of course, I mean, you will know, and the listeners hopefully too, but I'll put it out there as a disclaimer. It's not that simple. There are still glitches in the system, of course, but in terms of sample access, data quality, and just the depth of the phenotype data that's available here, there's not a lot of countries. There's some in Europe, but that's pretty much it. And the point is that until now, it's not really been delved into. The samples are just there because or not we, but people have collected them, and you don't want to destroy them. But epigenetics and things that we can do now still relatively new, and they know that. 10 or 20 or 30 years ago. So the samples are just there. And it's exciting that we get to go back and use them.
Very, very. And maybe that speaks more volumes about the US healthcare system than it does our biobank infrastructure. So we won't get into that, but that's, maybe. Yeah. Um, so no, it's great to learn a little bit more about your background and everything you're doing. So let's zone in on your research that you've done with epigenetics. You say a lot of times, the ability for epigenetics to do things for epidemiology, and I just think that's great. So what can epigenetics do for epidemiology?
Yeah, so a key component in epidemiology, like I said, is the human, and we are biased and we are fallible. And so when we're doing investigations that include people, we rely on questionnaires a lot. We ask them about their alcohol consumption and their smoking and physical activity, and we want to measure their blood pressure and all these things. Um, and there are a couple of problems when you're talking about questionnaire data. People don't remember. People think you have an agenda and they try and tell you something that they think you want to hear. Or if you bring it into a global context, people speak different languages. People, or not all people, smoke the cigarettes that you buy in the supermarket. Some make their own or use a different kind of tobacco and so on. And so epidemiology is not one size fits all. And if we're talking about really increasing diversity in epidemiology, we need to find biomarkers that are more one size fits all. And omics and epigenetics can do that. If we find a way, like there's been progress in this direction, if you find a way to accurately quantify lifetime exposure to certain things, to pollution, to smoking, to alcohol, then we don't need to ask people. We can still ask them, but we don't need to fully rely on that. So I think that is key. And the second thing is, if you link it to the biobank aspect, is we've got someone with a liver sample or blood sample from 30 years ago, and we didn't think at the time to ask him about specific things that we know are important now. But if there's DNA in that sample, which there will be, then we can measure certain components and we don't need to ask him or her. So it's powerful. It's something I get really excited about because you are able to look at these phenotypes in an unbiased way. And you also take one sample from a participant and you can get thousands of measures. And I don't have to say, okay, can you come back? I need another blood sample and another one. And I can just take the same DNA and I can read so much into it.
Yeah, that's great. So, and I want to stick on that topic as well. And you would need another sample if you want to look at them, longitudinally down the line, right? You wanted an update there. Okay, perfect. Just wanted our listeners to say, wait a second. No, that's great. So if you investigated my epigenome using a blood sample or buccal swab or whatever tissue that you had, even one that you just mentioned in the archive biobank, what are some things that you could just tell me about myself? Kind of in terms of what you've investigated so far.
Yeah, so like I just mentioned, we can say something about your alcohol consumption. Not as accurately as we can with smoking. With smoking, we can really tune in to exposure overall. With alcohol, we can see whether you are a frequent consumer or not. There's some nuances in the kind of alcohol that you're consuming and differences in consuming one drink every day versus only 7 drinks on Saturday. But I can tell you if you're consuming alcohol or not. I can tell you your age. chronological and biological. Uh, as I know, you've also discussed on this podcast the whole idea of biological aging or epigenetic aging. Um, in some instances, we can look at trauma, exposure to trauma. We can look at risk of future diseases. We can identify or stratify between current disease subtypes. And again, all of this, and you will know this probably even better than I do, but this is not in the public space yet. I don't think you can walk into a doctor's office and say, here's my epigenome, tell me everything you can about me, because things are moving at such a rapid such a rapid pace, and we are just improving these biomarkers and really trying to figure out what works for who and what do we actually see and capture in these biomarkers and how reversible they are. So we can tell you a lot, but with a relative confidence interval.
Yeah, it makes me so excited as well. I was talking to someone previously in my show, and they stated, and I'd love to get your opinion on this as well. You know, there's not a stronger signal between anything than there is between methylation and smoking, right? Like you said, it's better than what you're writing down on a paper survey, essentially.
Yeah, it is. It's quite incredible. Yeah.
Yeah. Well, let's dive more into the work that you do in particular. So I know you focus on researching understudied populations, and that's just amazing. I think that's, very much needed in this space. And there's probably a rather obvious answer to this question, but I want to ask you from more of a professional view who, dedicates their life to this type of work. Why is this important? What can we learn from these low and middle income countries? What is the research community missing out on by not looking at these populations?
Yeah, I think that's a really good way to phrase the question, because as someone that used to be a researcher in those communities, there's often this idea of, oh, it's the right thing to do to also include these understudied populations. But actually, I mean, yes, it might be the right thing to do, but it's also the smart thing to do from a researcher's perspective. The more diversity you have, the better your data set will be. Now, of course, if you have a limited ability and you can only get a small number of samples, then you want to try and limit diversity, because otherwise you won't find any signal. But where we are at this stage is really at a place where we can start including more and more and more diverse samples. And the research I did was in a specific South African population. And the complexity of something like smoking, we say there's such a strong association here. What about people who need to make a fire in their homes to cook, or people that work in a mine, or like, what is smoking? Is it really the cigarette? Is it just the inhalation of smoke in general? Is it the nicotine? Things like that you can only start really disentangling when you look at communities that doesn't smoke in the white European way. If I can, put it as simply as that. And we are dealing now with health problems of that's been in the making in the past 10, 20, 30 years. That came with urbanization and how our lives changed, and we see the consequences of it. And there's a part of the world where the process of urbanization is only just beginning or is still happening. And so if we want to understand some of the diseases that we're dealing with in the Western world at the moment, it would be quite wise, I think, to go to places where it's still happening and actually investigate it as it's happening. Um, so there's a lot. I saw earlier in the week a study. It wasn't published earlier in the week, but I only saw it earlier in the week, looking at ethnic differences in epigenetics and in DNA methylation, and more than 70% of the variation was environmentally driven and not genetically driven. And I think that is, I can't really say it is surprising, but it just goes to show that it's not enough to say, okay, but we have African genomes. What about the exposures? What about the lifestyle? I think that's really crucial.
And I think looking at those understudied populations before it's too late, right? A lot of these may, again, I don't know the right word here, but like disappear, I guess, or some of these patterns or things, it's just going to be too late. So we need to pay attention. That way we have a reasoning to take that more targeted, approach, preventative approach, if we want to move this and actually push this into, doctors' offices or make it commercially available for people to learn more about their health.
And I mean, on that note, the original idea for my PhD work was to look at or to compare a rural and urban population group in South Africa and then look over 10 years. So we had them at an examination in 2005 and then again at 2015. And I went to the rural site where data was collected in 2015. And this is really like gravel road, riding a donkey, um, slaughtering our own animals. There's one kind of kiosky thing that you can buy some food from, and that is the lifestyle. And even there, we were rather disappointed to find that it is already too urban. It is already too close to urban in terms of exposure, in terms of smoking, alcohol consumption, BMI, in the South African context. And of course, you have a problem there because in rural communities, you have people traveling to urban sites for their jobs. And so you can't really disentangle that. But we really thought we would see differences, and we couldn't. It's already too urban. So you're right. It's the clock is ticking, and if we want to know more, we need to do it now.
Yeah, exactly. And that was just to summarize, that was your urban and rural divide study that you performed. And that was over a 10-year period where you were looking at kind of these complex changes in environmental exposure and, rapid increase in non-communicable diseases. Was that the study you were mentioning?
Yeah, so that was the original proposal. Once we saw that we couldn't actually perform the quantitative investigation of it, we wrote quite a large review, starting from what we wanted to do and then also just looking at how it could be done better. And then we focused some more on the study population that we had, but we couldn't follow them longitudinally. So the review that I think you are referring to, that contrasts or that talks about the value of the urban-rural divide, was a summary of the available literature and then looking at what studies are going on at the moment and how they can be used. Yeah.
Yeah, that was the review I think I was referring to. So yeah, nice to understand that a little bit. I know a lot of your work backing up some with the DNA methylation is going to be looking at just that and then the association with those non-communicable diseases. So I'd love for you to define that just for our audience to link, that in their head. And then, yeah, we'll start there.
So non-communicable disease is just a disease that is non-infectious. So we're talking about your heart disease, your cardiovascular diseases, cancer, diabetes, dementia, Alzheimer's disease, those kind of diseases. And we typically associate them with environmental changes, with stress, with diet, physical activity, sleep, those kind of exposures. So it is not infectious like your HIV or tuberculosis or so on. So yeah.
Thank you for that. Just clearing that up there. And then my second half of that question is going to be, I guess you can decide how broad term or how far in you want to go here, but what is the typical association you see between DNA methylation and those communicable diseases? And NCDs is what I'm going to say now. A tongue twister. So yeah, if you want to explain that a little bit.
Yeah, I whenever I teach classes on epigenetics, especially with epigenetic aging, I tend to start the class by asking people, what do they do to keep themselves healthy, or to, what are the good habits and the bad habits? And it's always something like, I try and sleep 8 hours, and I try and just not take the elevator and use the stairs, and I try to not smoke, and all these things. And then we go into the class and we look at the different effects that these exposures have on DNA methylation and on things like epigenetic aging. And then we look at the downstream effects of those. And my final slide is always something like, it turns out your grandmother was probably right. Like, you should do all these things. We didn't know why. We just kind of followed her. Or, it's typically in my head, it's the grandmother figure, but whoever it is, where the wisdom comes from. And it seems like the more we move forward in this field, it just backs up all those, old wisdom. So we see that obesity influences DNA methylation, mostly in a way that increases your risk for hypertension, type 2 diabetes, cancer, and so on. We see that obesity associates with pro-inflammatory profiles. We see that diet, an unhealthy diet, relates to unhealthy lipid profiles, and that relates to increased disease risk. So it mostly makes sense, I would say, and it's in the direction that you would expect. But so far, we've seen these associations, and we don't really understand how the body responds in the way it does. And omics, going back to the omic epidemiology enthusiast, the omics really just provide some of that substance to what we've observed for so long, and to, what our grandmothers told us to do from the start.
Yeah, I picture a grandmother figure in my head too, for what it's worth. Yeah, no, and you're actually teaching this stuff then, right? You're teaching this to students and um, classes? Yeah, because I think I maybe had like 30 seconds. Hey, this is what epigenetics is, right? And like maybe a biology class, but that's amazing to hear that you're actually teaching this. And yeah, and hopefully informing them on future career paths in this work or different research at least. Mhm. And I'm just going to move through some of your other studies here that I'd love to hear a little bit more about, Dr. Cronjé. I know you did one on the comparison of DNA methylation clocks in Black South African men. Can you chat about this study and maybe what you found and give us some insights there?
So we did the study as a way to compare the different clocks that we know work really well in predominantly white, predominantly European ancestry samples. We wanted to see if they perform as well in our cohort. And overall, we saw that our cohort was consistently younger epigenetically than they were actually chronologically, for all the clocks. And we strangely observed that the Grimm age clock, which is actually not a measure of age, it's not like your Hannum or your Horvath clocks where you just get an idea of what someone's biological age is. It's more about what is your risk relative to your age. So you have the risk of dying of a typical 60-year-old, for example. And then we know, okay, but you are 55, so this is a problem. So that is how the Grimm age works in a simple way. And the Grimm age gave us the most precise age estimate, which was interesting. The rest really underestimated. And when we really delved into the components of these different clocks, we found that the Grimm age probably performed particularly well because of the high prevalence of smoking and alcohol consumption in our study group. Much higher prevalence than in some of the European samples. And this is the only clock, it was at that stage, the only clock that incorporated lifestyle aspects to the clock. So it took a methylation, or it still does, it takes a methylation component that reflects lifetime exposure to smoking, for example. And we think that a lot of what drives aging in this population is the exposure to smoking, be it tobacco, cigarette smoking, or, like I mentioned previously, having to warm yourself with a wooden, fire in your home, or cooking on fire, or working in a mine, or so on, instead of things like obesity and unhealthy diet, so on, because that is not a problem that we have in this particular cohort. They are all fairly lean, fairly fit. They do physically active jobs. And it's a carbohydrate-based diet mostly. So it was an interesting study.
Yeah, definitely. So just to clarify, they had higher Grimm ages, you said, or lower Grimm ages?
So their Grimm ages were higher than the other ages, but it was the closest to chronological age. So it's the one with the least discrepancy from actual age, which is not necessarily what you would expect, given Grimm age's goal, but that's what we found.
No, it makes sense. Wanted to ask there. You also looked at some epigenetic age acceleration in the cardiometabolic disease among migrant and non-migrant African populations. So, yeah, I thought that was super interesting too. So, what did you find in that one?
Yes, and in this study, we served, or our cohort served as a replication cohort. And the main focus of the study was looking at a population from Ghana. One group of the cohort remained in Ghana and one migrated to Europe, Amsterdam and England. And we saw differences in the way that the migrants and the non-migrants age. So we found that the Ghanaians that migrated overseas had an accelerated aging, and those that stayed in their community did not. And then within each community, we also looked at some dietary factors. So there was an association with increased folate consumption, so vitamin B9, and decreased aging. But then also there was an association between higher BMI and slower aging, which was unexpected because usually we see obesity increases or accelerates age. And in the paper, there's quite a lot of discussion about how in these communities, having a higher BMI could be an indicator of health, of socioeconomic status. I mean, we're not talking about obesity, but we're talking about having adequate reserves, having food, having, protecting yourself from other stresses by having a BMI that's a bit higher. So we're not advocating for higher BMI, but in these groups and in our cohort in South Africa, we saw the same thing. BMI was protective.
Yeah, very good. No, interesting to say the least. And I'll make sure to link out all of these studies. So if someone wants to look a little bit more in depth or read it, they're very great reads. I do recommend looking into them. But you're giving us great summaries of those. Another published, it was more, I guess, of an editorial that I found of yours. That was done, I think, in late November of 2021. But this was titled, Could Unlocking Methylation-Based Blood Cell Counts Revolutionize Epidemiology? And we are doing a lot of work with immune cell subsets right now. And I just think you really, jumped on that idea very, very early. So can you summarize what you wrote in that excerpt for our listeners?
Yeah. So the idea for that came from one of the papers that was part of my PhD, where we were looking at blood cell counts using methylation data and comparing that to other inflammatory markers, so CRP and interleukin 6 and TNF alpha and so on. And we observed that these relative blood cell counts, so your neutrophil lymphocyte ratio, for example, just outperformed the conventional biomarkers of inflammation. And if you look at the literature, it makes sense that it does. It does give you a better idea of relative immune activation. And it is used in the clinical setting often. And so I started just paying attention to the literature and in papers that I read after that. And it's just not something we see in epidemiology, that people incorporate blood cell counts and adjust for it. And as it turns out, the reason for that is because you need to do that in a fresh blood sample. That's why we can do it in a hospital. We take your blood, it immediately goes to the lab. They can do these analyses with it. Whereas CRP, interleukin 6, and those we can do in frozen samples later. So we can collect thousands of samples for a cohort, and then years down the line, we can look at those inflammatory markers. And the exciting thing about DNA methylation is we can do the same. So we can look at a sample from 10 years, 20 years, 30 years ago, and actually quantify relative immune cell proportions in those samples. And so the idea of that editorial was just to say that if we know from an epidemiological point of view that these things matter, that blood cell count is an important confounder to adjust for, and it's an important predictor of outcomes, why wouldn't we start including it? I would think the reason is because it sounds really scary. Like, oh, but then I need whole genome DNA methylation data, and it's millions of CPG sites. And the point of the editorial was just to say that at the end of the day, after you do your quality control of the DNA methylation data, you get 5, Excel columns that tell you neutrophils and lymphocytes and so on. And it should be no different than having that Excel column of CRP or interleukin 6. And we need to communicate that better to the epidemiology world. And when we have huge cohorts that also have methylation data, it is worth informing researchers and say, I will send you the data set with CRP and IL-6. Would you also like to have these methylation measures, epigenetic age, an indication of smoking, those kind of things? And yeah, we should use those because they're valuable.
Just that's another piece to the puzzle, right? Some further insights that you can learn. So then we'll start to unlock kind of things we don't know what we don't know essentially. So trying to learn a little bit more. Well, we have a couple more questions for you here, but we are getting to the end. Besides people not giving enough attention to these understudied populations, what are some other challenges in your work that you face or that you think is a big concern? We kind of just mentioned one, talking about sharing the data and making this obvious as a resource to the epidemiologist. But is there anything else?
Yes, it's something we've been struggling a lot with. It's just the tissue specificity of it all. Um, it is an incredible resource to have a biobank, of course. But what does methylation of the liver tell us versus the kidney versus the pancreas, versus blood? And so ideally, you want samples at the same time point in the same individual that you can compare, and we can figure out um, which tissue is better for which disease and so on. But we don't have that because it's unethical, and people don't want to just give you all of their biopsies. But also, there's a bias in biobank samples because there's a reason I go for a liver biopsy. I don't do it for fun. Um, and the reason is most often that I am ill or something has changed or we saw something on a scan. And you can try and narrow down your search by saying, I only want the people who went for biopsy and we know that they didn't have cancer or whatever they were looking for. But there's still a bias in that. So I think the next challenge for both the omics world, but also the epidemiology and methods development world, is really to try and quantify the bias in samples like these, and better understand who goes for these biopsies, um, and how can you best use the tissue and adjust for the confounding by indication bias.
Yeah, yeah. No, that's a good one, and hopefully one that will be addressed as we have that full profile of the person, or know why they were in there, and why their samples are in that biobank. So I appreciate that answer there. I'm excited to ask this next question. I know you haven't published in epigenetics in a year or so, which is not that long at all. Um, but you
Years, it's very long. The field is just running. In epigenetic years, it's very long.
oh, yeah, epigenetic years. Um, but you mentioned to me when we were chatting offline, you may pursue this epigenetics-based project within the next, few months or so. And I don't know anything about it yet. So would you be willing to share some insight about that project, and maybe what's next?
Sure. So it is within these biobank samples, and once you've generated the data, you get into that fun phase of writing up and doing the stats and all that. But before you get to that point, and what I've been doing in the past 2 years is figuring out which samples we want. What is the question that we want to ask? How are we going to ask it? Getting the biopsies from the biobanks. And separately from that, I also got a grant from the Alzheimer's Association. So I'm focusing a bit on dementia. And the grant was for proteomics, for new proteomics analysis, but the goal is to do a multi-omic baseline profile of these participants and then really look at what we can say about risk in the future. And these samples are also part of clinical trials, with different outcomes, but with interesting treatment. So the idea is also to look at treatment effects and if we can stratify participants based on who will gain most in terms of cognitive function in the long run, or at least a delay in cognitive decline in the long run, based on these treatment effects.
Yeah, well, congratulations on the grant. That's very big. I know that's not easy. Are what are you treating those groups with? Am I able to ask that? Or
Yeah, so this is all again the gold mine of epigenetics. These are old trials that have samples. So in the US, there's an incredible resource that I only learned about 2 years ago, BioLink. So that is all the NHI, NHI, National NIH. Yeah, sorry, all the NIH funded projects that have samples left get stored by the NIH, and you can apply for access to some of those samples if it fits within the original scope. And that is really exciting because if you've got a trial that looked at blood pressure reduction, which is the one that I'm working with, you can now take those samples, look at follow-up data without needing to do a new clinical trial and asking people to give new samples or go through the process again. So when we talk about what can epigenetics bring to epidemiology, I think it's this. It's that we have so much data that we haven't even started looking at.
Absolutely. Well, that's great, and I'm excited to keep up with all of your studies in the future. I have one last question for you, Dr. Cronjé. It's a curveball question, so nothing related to your research. But it's just kind of fun. So, if you could be any animal in the world, what would you be and why?
Oh, um, any animal. Oh, I have to say a lion. And now I have a question for you. Have you ever seen seen a lion in a wild or in its natural setting?
I haven't. No, I want to do the safari. I want to, go to South Africa or Africa. I want to do it all, but I have not, and I'm sure it's beautiful.
Yeah, so they are just incredible. And they are maybe the reason I associate with them is the way that they can just sleep in the sun um, and just lay there and have the time of their lives. And then in the next moment, just get up and be so incredibly powerful and
Hmm.
roar in a way that just uh, feels like it's shaking the earth around you. Um, so that would be my answer, but that is a curveball.
A great answer. No one's that was beautiful. It has a great explanation. No one's given that answer before. So, yeah, I definitely want to go and see all those animals in the wild. So, Dr. Cronjé, we've come to the end of this amazing podcast interview. So, for listeners who want to connect with you, where can they find you? And I'll put this in the show notes.
Yeah, so I am most active on Twitter, I would say. So they can find me on Twitter. They can also connect with me on LinkedIn. And then if you go to the University of Copenhagen website and you search for me there, you will find all my other contact information there.
Amazing. Well, thanks everyone for joining us at the Everything Epigenetics podcast. And remember, you have control over your epigenetics. So tune in next time to learn more. Thanks, Dr.
Do what your grandmother told you. I think she's right. Yeah. Thank you so much.
Much.